Hello and welcome to GeneLab Chats. Today we'll be interviewing Dr. Amanda Saravia-Butler from KBR and the NASA Ames Research Center. Dr. Saravia-Butler and colleagues used several GeneLab datasets to identify technical variations that pose challenges associated with making comparisons across datasets and also introduce a new approach for identifying and correcting batch effects when combining GeneLab RNA-seq data. In this interview, Dr. Saravia-Butler describes his research and how the GeneLab data systems and analysis working groups contributed to this work. Thanks for joining us. Could you please let our audience know the most important result of this work?
Hi, thanks so much for having me. Yeah, sure can. So there's often a need to combine data, such as RNA-seq data, from multiple experiments to enhance statistical power. However, doing so could introduce technical variations, or what we refer to as batch effects, that may overwhelm or hide any true biological signals from the combined data. And although there are several computational methods that have been developed to correct for batch effects, after reading through the literature, there didn't seem to be an ideal method or a clear means to quantitatively evaluate the different methods available to determine which would be the best one to use for a particular set of aggregated data. So I would have to say that the most important result of our work was that we developed a method to do just that. The method we present in this work provides a means to first assess a set of aggregated RNA sequencing data to identify the most prominent technical variable or variables among the aggregated data, and then to evaluate the ability of 5 commonly used methods to correct for them using a set of quantifiable criteria. We then developed a custom scoring approach that uses the metric from those evaluation criteria to identify the optimal correction method to use for the specific combined data set. And although we demonstrate our approach on a space biology use case, our approach is actually broadly applicable to scenarios in which multiple RNA-seq data sets are combined, thus introducing technical variation.
Thank you. And the second question is, how did the GeneLab data systems or the analysis working groups help assist you in your work so you could achieve these goals?
Yeah, thanks so much for that question. So this study actually would not have been possible without the Open Science Data Repository, GeneLab, and the analysis working group members. So the Open Science Data Repository, for those who don't know, provides public access to data from spaceflight missions. And since spaceflight missions have the issue of low sample size and therefore often requires the need to combine data from multiple experiments in order to enhance statistical power, data from these missions serves as an ideal use case for this work. So by searching the repository, we were able to find 7 liver RNA-seq datasets that were derived from multiple rodent research spaceflight missions to use. And each dataset was comprised of rich metadata, allowing us to clearly identify the technical and biological differences between them. And furthermore, since GeneLab, which is part of OSDR, processes all RNA sequencing data sets that are hosted on the repository using a consensus processing pipeline, we're actually also able to download the publicly available processed RNA sequencing data from the 7 selected data sets to use these in our own study without having to consider any technical artifacts that could have been introduced if these data had all been processed differently. So thanks to OSDR, we had an ideal set of combined data to use for our study. And then the analysis working group members come into play when we realized that we really needed statistical expertise to help us develop an unbiased approach to quantifiably compare the results of the evaluation criteria for each batch variable correction method pair we investigated. So we presented this issue at the monthly OSDR AWG meetings, and Hamid Chok, a statistician, active AWG member, and the second author of this paper, quickly volunteered. And Hamid was able to use his statistical expertise to develop the custom scoring approach used in this work to identify the optimal correction method for a given set of combined data. So really, this study wouldn't have been possible without the unique expertise of our analysis working group members, plus the data available on the Open Science Data Repository.
That's amazing to hear about the collaborative work. And thank you for sharing your time with us and our viewers. And to our audience out there, you can learn more about this work, how to join the analysis working groups, and access all the data sets analyzed in this work with the links provided in the description section below. Thank you and have a wonderful day.
Thank you.